Welcome to Discover ASAP, the video interview series where we highlight the top findings and tools connected to the ASAP network. Today I am talking with Sara Bandre, s-Ciga, head of Neurogenetics at the Center for Alzheimer's and Related Dementias at the National Institutes of Health, about the NeuroBooster Array, a genome-wide genotyping platform to study neurological disorders across diverse populations. Sara, to start, could you tell us briefly about your overall ASAP project and the impact you hope to have on Parkinson's disease?
Yeah, absolutely. So the NeuroBooster Array, or NBA, as we call it, is the core genotyping platform that is used currently at the Global Parkinson's Genetics Program. It's an ASAP-funded initiative that aims at democratizing and diversifying genetic research in Parkinson's disease. GP2, the Global Parkinson's Genetics Program, aims to support the genotyping of over 200,000 individuals from a broad range of ancestries to accelerate discovery of the whole etiological risk spectrum. So when it comes to both common and rare genetic risk factors associated with Parkinson's disease and also atypical Parkinsonism. And this is mostly focused especially in historically underrepresented populations. And by integrating global data and fostering open access collaboration, GP2 goal is to uncover novel biological insights into the causes of Parkinson's and really contribute to the identification of potential therapeutic targets. Ultimately, what we aim here is to support the advancement of precision medicine in Parkinson's disease and ensuring that future interventions are relevant and effective across the diverse global population spectrum.
Wow, that's a really ambitious goal, so it's great to hear about it. Focusing now on your work, could you give a brief overview of the NeuroBooster Array, or NBA, and how it works?
Yeah, so MEA is a custom genotyping platform. So this was specifically designed to support large-scale research into neurological diseases with a major focus on Parkinson's disease, also Alzheimer's and other relevant conditions. So genotyping arrays like this one are very key for our field because are going to allow researchers to scan hundreds and thousands to millions even of known positions in the genome that are going to detect genetic variation, right? And so when we pair with the genome-wide association studies, this data is going to eventually help us identify genetic regions that are more common in individuals that are going to develop disease compared to those individuals that do not develop disease or controls, as we call. And this offers very important and valuable clues about the genetic basis of the disease, which is really the foundation to understand the etiology of Parkinson's disease in this instance. The NBA combines broad and global utility in terms of the whole spectrum of neurological disease-specific content. It includes about 1.9 million variants from the Illumina Global Diversity Array. Together with custom content, it's about 95,000 additional variants that are related to about 70 different neurological conditions, including Parkinson's, plus about 10,000 tagging variants to enhance imputation accuracy across ancestry. So essentially in the lab, when we obtain high quality genomic DNA that is going to be enzymatically fragmented, we are going to hybridize the DNA to the array using automated systems like TCAN workflows, and then eventually we'll scan the chips. And the resulting data that we generate through the chip is enhanced through imputation with reference panels such as TopMed that is going to allow researchers to infer millions and millions of additional variants with high accuracy, especially when it comes to diverse populations. And this is what makes our chip really a powerful tool for the future, not only for variant discovery, but also for variant validation. And it really provides globally relevant insights when it comes to neurological diseases like Parkinson's.
Yeah, it sounds like you're really filling a gap in the current research landscape with this, which leads nicely into my next question. And you touched on it a little bit already. So feel free to take this in, whatever direction makes the most sense for you. But why did you create the NeuroBooster Array, and what advantages does it have over other existing tools like it?
Yeah, absolutely. So, well, this was a team effort, mostly led by Dr. Mike Knowles and Dan Vitale. And we created this because previous existing genotyping arrays, such as, for example, NeuroChip or NeuroX, were heavily biased toward European ancestry. So, they were not capturing all the genetic variation that is important for us to understand disease globally. And so, this offered limited ability to further dissect the genetic architecture of disease across all the populations, right, to understand the whole picture. And so the chip was designed to expand ancestry, diversity of genetic studies, to provide more deep neuro-specific content, including rare and low-frequency variants, and also to improve imputation quality. That is really, really important when it comes to further dissecting risk and fine mapping all the different regions that we find associated with disease across ancestries.
Okay. Yeah, that makes a lot of sense. I'm excited to see it, what it produces. And leading on to that, how do you intend to use the NeuroBooster Array? Are there ongoing projects or any ideas to further our understandings of Parkinson's, or how might other researchers use it?
Yeah, so this chip really serves multiple research strategies, and the Global Parkinson's Genetics Program is releasing data every 6 to 8 months. So that is really valuable. First of all, we have to understand that all high probability monogenic Parkinson's disease cases always undergo genotyping with the NeuroBooster Array, and then for those unsolved cases, we are going to prioritize those samples for future whole genome sequencing efforts. So I think for monogenic is quite important, but also for complex disease, because this is going to enable large-scale GWAS across diverse populations, and is very important to keep improving, imputation efforts for future fine mapping. The more samples we have, the better picture we are going to be able to make about the etiology of the disease. So definitely for discovery and for recruitment, discovery of identifying pathogenic variants to help prioritize samples for sequencing, and even in the future, who knows, clinical trials when we think about patient stratification efforts. In general, I think for global collaboration is going to be key. Researchers anywhere in the world can genotype samples using the NeuroBooster Array. This is available for the whole scientific community that can join existing GP2 efforts or conduct independent analysis, even supporting, again, disease mechanism discovery and future therapeutic approaches.
Yeah, that's super exciting. I can definitely see how this will enable a greater collaboration across the globe, and enable more people to take part in Parkinson's research and benefit from it. And related to that, also, how could someone access the NeuroBooster Array for their own lab? Is it commonly available? Are there any tips or tricks that you might need in using it?
Yeah, so the NeuroBooster Array is available through Illumina and is currently being distributed through the Global Parkinson's Genetics Program. Illumina is the supplying system, is going to supply our custom array with our custom content and all the pipelines. So I will say interested labs need to join GP2 or partner via ASAP supported efforts. That will be really positive to keep increasing the sample size of this massive initiative. They can submit an application or a sample proposal through GP2. I encourage them to check our website and to get in touch. The team is delighted to help everybody, and then post-genotyping results are always deposited into the GP2 cloud platform for analysis by both submitted groups and the broader research community. And for those that are out of GP2, I think collaboration or sample setting arrangements may be possible through ASAP or related working groups.
Okay, great. Is there anything else that you would like to share that we didn't ask about so far?
I just wanted to highlight that MBA is an amazing effort. It not only helps in terms of variant discovery, I think also ensures robust cross-ancestry imputation, improving inclusivity in genetic research. That's a very important point to make here. It has already led to important findings. For example, I encourage the scientific community to check our last African GWAS, in which we unravel a novel genetic risk factor in GBA MBA. And, the platform supports open science. We have protocols, data, and code that is openly shared on GitHub and Zenodo and structured publicly via our GP2 portal. So a lot of resources out there to keep learning about our field.
Okay, great. Yeah, we will try to put all those resources in the show notes. The paper that describes the NeuroBooster Array, the paper that you just mentioned, how to get involved with GP2 and ASAP if anyone is interested. So yeah, hopefully we can continue building this effort. So it was really great to hear more about the NeuroBooster Array. And so thank you so much for your time and for chatting with us. And we look forward to hearing more from you and GP2 in the future.
Thank you so much.